Hello, Dr. Johnson's sections of Introductory Psychology Psych 101, Lincoln University of Missouri. I'm recording this for us on the afternoon of Saturday, April 11th at just about 1:40 in the afternoon. And in this presentation, we will be going over the individual parts of the neuron and what they do. So if you have printed out this handout and want to make notes, or if you just want to refer back to this slide that is the same information in a slightly different visual format, you can do that. In the previous presentation, in Lecture 1 for Unit 4, we talked about basic concepts, sort of basic facts to know about the neuron. And now we're talking about the individual parts and how they relate to the overall function of the neuron. We start at the head of the neuron. We move on to the tail. We're going to, in this presentation, we'll fill in all of these blanks. We will leave the detail for blank D for the next presentation, which is about the action potential. I've continued to experiment with the size of the box that translates from what's on my screen to your screen. I hope the editing is a little less choppy today than it was in the last presentation, but we'll see. Okay, keeping in mind that in the first presentation, one of the things that I suggested to you is you think about the neuron as being like a machine. And so now we're talking about specific parts. And the first part is these twisty branches that grow off the cell body of the neuron. And these twisty branches are called the dendrites. And the most important thing to remember about the dendrites is that the dendrites have receptor sites to receive neurotransmitters. The neurotransmitters are the chemical keys that we talked about in the previous presentation. One of the things that is, I think, kind of obnoxious about the vocabulary that we have to work with when we're talking about this material. There's really two things that I think is kind of obnoxious. One is that the vocabulary is more jargony, it's more complex than I think that it actually needs to be. And partly that is the result of the fact that a lot of this stuff was first spelled out, laid out in the last 25 to 30 years of the 19th century, when in the field of medicine, the predominant languages, you still were expected to know Latin. You were probably expected to know some Greek. Even if you were speaking in English, you were expected to know some German. So a lot of the terminology that we have when we're talking about this comes kind of from this perspective that's a bit antique from our vantage point today, but it's established and it's the language that we have to use. So for anybody who is, for instance, in pre-nursing or pre-med, and you come across, or you're going to major in psychology, and you will come across this material at increasingly deeper levels as you progress through your education. You'll find this vocabulary again, and you'll also see some variations on this vocabulary. So anyway, the word dendrite comes from the Greek word for branches. And to me, this seems kind of random because it looks to me like when we look at the overall neuron, you've got something that you could reasonably call branches at both ends. But we use the Greek word, the dendrites, for the branches that are at the head end. And if you kind of want to remember a mnemonic, what is mnemonic? A mnemonic is a short phrase that you can remember easily that helps you identify sort of a deeper point. Dendrites detect. What the dendrites do is they receive the message in the form of the chemical keys, the neurotransmitters, and that message is going to be either turn on or stay off. If we're looking at the class handout, we've got two different blanks. We've got B1 and we've got B2. So B2 is the cell body. This is the main structure of the individual cell. And one thing, there's really two things to remember about the cell body. One is that if the neuron generates its electrical signal, the action potential, that starts at the cell body. And the second thing to remember is that the cell body contains what's B1 on the handout, the nucleus. So if you've had biology classes and know stuff about cells, or other science classes where we've talked about cells, every cell in living organisms that we know of, whether they're animal or plant, I don't know if this applies to the fungi, has a nucleus. And one thing to remember, an important thing to remember about the nucleus for our purpose, is that the nucleus contains the instructions, the genetic instructions, both for how to build the neuron, how to make the cell that we're talking about into this particular type of cell, the neuron, but also what neurotransmitter chemicals to produce. So throughout this unit, we're going to be talking about neurotransmitter chemicals as being chemical keys that fit into locks. You may not know it yet, but in that video that we looked at, yes, in the previous presentation, and that we'll look at again later in this presentation, we've already seen the chemical keys at work, and there are a lot of different chemical keys that our nervous system produces for a variety of reasons. And the nucleus, part of the genetic information in the nucleus is, okay, you particular neuron, you're going to build one specific chemical key. So, you know, I encouraged you in the previous presentation to think about the neuron as being like a little machine. And if you extend that metaphor, the neuron is a machine. And you think about different kinds of machines that we work with, whether or not it's our desktop computers or our cell phones, or for instance a car. What happens when you open up the hood of a car? you look under the hood and you see separate components, and all of those components are necessary for the car to do everything that the car does. You're going to see the engine block. It's going to have the pistons. You're going to see the cooling system. You're going to see the electrical system. You're going to see some other stuff. So there are individual sub-machines inside of the neuron, just like if you pop the hood of your car, that do particular things. And I'd like to encourage you to think of the nucleus as one of these, and the specific function of the nucleus that we're paying particular attention to for our purposes is gives the instructions for what neurotransmitter to build. The other two sub-machines that we're going to be talking about as we explore everything that we're talking about in this syllabus as to how the neuron works are going to be the sodium pump and the reuptake mechanism. And one of the reasons that I think that it's helpful to think about the neuron as a machine is, okay, think about machines like your car or your cell phone. In order for those machines to work, they have to access energy. They have to burn fuel. You have to charge the battery with electricity in terms of your cell phone. You have to have gasoline in terms of your car. And one of the things about our individual submachines is that in order for them to do their job, they have to access some kind of fuel. They have to burn energy. We'll get back to that. Okay, so we've got the dendrites here, we've got the cell body, and then in particular we've got the nucleus. And leaving the cell body is one, this is the main part of the tube, so blank 3 on our handout is the axon. So one axon leaves the cell body. And sort of as generalization about neurons in general is that any given axon, even though only one leaves the cell body, it can grow an indefinite number of branches, twigs at the end. And each of those twigs reaches out to talk to either a separate dendrite on another neuron, or if we're talking about the neurons that give instructions for our muscles to move, a piece of muscle fiber. So a second mnemonic device that is offered to you is axons away. The main purpose of the axon is to carry a message, and within the neuron it's going to be in terms of what we talk about as the action potential, a brief generation of electricity away from the cell body to what is labeled as E on our handout. So like a lot of axons in the nervous system, the one in this illustration has several branches, and you'll see that each of these branches ends in a little sort of bulb shape, and those are the axon terminals. And as I mentioned earlier, sometimes if you continue to have to study this material, you'll sometimes see it called axon terminals, sometimes you'll see axon terminal bulbs, axon terminal buttons, axon terminal boutons, in a couple of the videos that we'll look at. But for our quiz language, we're going to talk about them as being axon terminals. And the axon terminals, the axon terminal bulbs, axon terminals. So here's the second part of our illustration, the cutaway in the handout. That's this box. So we see that the axon terminal, and we saw this in the video, has these spherical packets. In the video we were looking at, they contained red dots. In this illustration, they contain yellow dots. This slide just talks about the fact that if you continue this, you'll find alternate terminology, but it's all talking about the same stuff. Those spherical packets that are hanging out in the axon terminals are called synaptic vesicles. On our handout, they're the blank F. Again, sort of referencing the fact that we're using vocabulary that I think is unnecessarily complicated. It would have been easier to call these spherical packets synaptic vesicles. It would have, pardon me, easier to call them synaptic vessels, but they didn't ask me 100 years ago when they were finally getting microscopes with enough power to be able to look and see how these things were made. In this illustration, the synaptic vesicles don't hold yellow dots or red dots, they hold black triangles. And depending on the illustration that we're looking at, those red dots or the yellow dots, the black triangles are representing the fact that the vessels contain neurotransmitter chemical molecules. Neurotransmitter chemical molecules. So on the handout, those are labeled H as the black triangles. So when an action potential reaches the synaptic vesicles, the action potential, the electrical current that begins at the head of a neuron and travels down the axon to the axon terminals, where the synaptic vesicles are hanging out, it pushes those bundles that are the synaptic vesicles up against the cell membrane. This pushes, okay, where do we get the name? What do the synaptic part of the name synaptic vesicle refer to? And so the action potential pushes the neurotransmitter chemicals through the cell membrane and out into what's labeled G, the synapse. The synapse is a very tiny gap between the axon terminal and the dendrite of a receiving neuron. So the dendrites, the way the neurons are built, so here we've got the dendrites on the head end. Here we've got the axon terminals. What this shows right here is that the axon and its axon terminal reaches out and comes really close to, but does not completely touch, the head end, the dendrite end of the next neuron, the neuron that's going to receive the message, or maybe muscle fiber. So here's H, neurotransmitters, and just once in the handouts, I spell it out completely, neurotransmitter chemical molecules. And there's a reason that I use all three of those words. Synaptic vesicles are located in the bulges, the bulbs, at the end of the axon terminals. The action potential pushes these vessels up against the cell wall and spits the individual neurotransmitter chemical keys out into the gap. Okay, what is, there's different ways to answer this question, but what's the major component that human bodies and animal bodies and plant bodies are made out of? What's the number one component? And the number one component is water. So, our overall skin, which our skin, by the way, is the largest organ in the human body, establishes the boundary of the body just the way the cell membrane establishes the boundary of the neuron. And so we've got all of these boundaries are different ways to contain ingredients that are mostly water. So one of the things that is helpful to keep in mind as we try to understand how it is the neurons do their communication thing is that it's like there's a soup on the inside of the cell membrane, and there's another soup on the outside of the cell membrane. And when the action potential reaches the axon terminal, it pushes these neurotransmitter chemicals, another ingredient, out into the soup on the outside of the cell membrane. And then some, but not all, of these individual neurotransmitter chemical molecules float across the synapse and bind into what is labeled on this illustration as H, receptor sites. So one of the things that I invite you to notice is that in this illustration, we're drawing the neurotransmitter chemicals as black triangles. In this illustration, which is in the PowerPoints, we're drawing the neurotransmitter chemicals as yellow dots. And then finally, in this animation, we're showing the neurotransmitter chemicals as these tiny little red dots. So in our animation, one thing that is good about this animation is that it shows that when these red dots, these neurotransmitter chemicals, find the receptor sites on the receiving end of the next neuron, these particular neurotransmitter chemicals cause these gates to open. Okay, so we've looked at neurotransmitter chemicals as being represented as red dots and yellow dots and black triangles. And none of those really are what neurotransmitter chemicals look like. Once the neurotransmitter chemicals float out into the synapse, some but not all of the neurotransmitter molecules lock into or bind to receptor sites on the receiving neuron. And so the receptor sites on the receiving neuron are illustrated as being I. Once the neurotransmitters bind into the receptor sites, they're telling one of two messages to the receiving neuron. And those two messages are either turn on or stay off. Earlier, we talked about the submachines in the neuron. The end of it, we said we're going to talk about three different gadgets that are subcomponents of the neuron. So the second one is the reuptake mechanism. And the reuptake mechanism reabsorbs any neurotransmitter chemicals that are in the synapse that after the action potential says spit these neurotransmitter chemicals out. Some of them don't bind receptors with receptor sites. And because natural systems benefit by being as efficient as possible, we don't want that material to go to waste. So the reuptake mechanism reabsorbs that stuff back into the neuron that spit it out. That doesn't really show up on our animation, but it's something that happens. End of this presentation, one of the things I'm really hoping that I can invite you to think about is that the shapes that our illustrations and animations show for the neurotransmitter chemicals are oversimplified. It is part of what I hope you'll think about in terms of like, how is all this stuff working in our nervous system and influencing our behavior and our experience, is that just as the neurons take up space, they have a material space, they got a shape, the neurotransmitter chemicals, and this is why I call neurotransmitter chemical molecules in that earlier slide, is to ask you to think about the fact that they do have shape. And shape is key to how all of this works. It does communicate. The way that our illustrations are drawn and the way the animation is animated is that both the neurotransmitter molecules and the receptor site have a unique shape. So let's go back and watch our video again now that we've gone through all of these the functions of these different parts. Let's go back and look at this video from the beginning. So we start with a kind of macro view, and we see these green lines wrapped around the brain. They're coming out of the brain, and the green lines represent nerves. And a nerve is comprised of many individual neurons that are bundled together. And throughout the rest of this unit, we're going to be talking about nerves and also neural pathways. There's the difference between the terminology nerves on one hand and neural pathways on the other is that nerves, for the most part, are outside of the brain proper, and neural pathways are inside of the brain proper. And one physical difference between them is that nerves are actually bundled together by fascia, by a biological sort of wrapping. So we zoom in on the nerves, and now the white flashy part is the action potential. Here is the cell body of a cell. Here's the axon terminal of the ascending cell that has formed a synapse with the receiving dendrite, which then generates another action potential. And now we see the red cross spheres go down inside the cell body and then immediately pop back out. That's all going to be in the next presentation. The action potential reaches an axon terminal, pushes the synaptic vesicles up against the cell wall, which releases neurotransmitter chemical molecules out into the synapse. They bind to receptor sites, and in this case, they send a turn on message and cause these gates to open. Now these little blue spheres enter into the cell membrane, and the fact that they are entering into the cell membrane indicates that they're sending a turn on message, because if they were sending a stay off message, then they would be encouraging the gates to remain closed. Okay, so the last time I was updated, which has been a year or two, we had so far discovered at least 145 individual neurotransmitter chemicals that are built by individual neurons in our nervous system. So that's at least 145 different keys, which implies that in terms of receptor sites, there are at least 145 different kinds of locks that the keys fit into. So the neurotransmitters, think of the neurotransmitters as keys and the receptor sites as locks. Because you can look at it with the naked eye and very quickly get what we've been talking about. Okay. Here's an electron scanning microscope picture of a particular neurotransmitter chemical that's produced by the brain. This neurotransmitter chemical was not identified until sometimes in the 1990s, so not more than about 25 years. And the scientists who originally discovered this neurotransmitter chemical called it anandamide. Here's a drug molecule. So this is not a molecule that our nervous system naturally produces. But you can look at it and you see, if you think about this as a key and you think about this as a key, it's like, oh yeah, they look close enough that I could imagine that this key, the drug molecule key, could fit into the lock that this neurotransmitter chemical key is built for. And this particular drug molecule is the drug molecule THC. So I'm certain, I would be surprised if anyone viewing this didn't already know that THC is one of the active ingredients in the plant-based drug cannabis, which we commonly sometimes refer to as marijuana. And in terms of how we are currently investigating the effects of cannabis as we go through our social experiment on a state by state level and a national level as to whether or not we're going to use cannabis as a medically sanctioned drug or a drug of recreation. There's two different things that at this point we've identified through research that active components in cannabis can do, and one of them is pain relief, and the other is alter our consciousness in a way that we commonly refer to as getting high. And so in the 1990s, when the scientists who identified, look, this is why cannabis works to get people high, is because we make this natural neurotransmitter chemical called anandamide, and the THC molecule fits into the receptor site for anandamide because it's got a key shape that is the same. Anandamide is from a Sanskrit word, and Sanskrit is a very old language that is associated with the Indian subcontinent and some of the oldest religious and philosophical writings that humans have produced that survived. For bliss. For those of you who have taken it upon yourself to investigate, or will take it upon yourself to investigate, what is currently established about how it is that the active components in cannabis can affect us. So as I said, there's like the short version is there's two different things. There's the painkiller part and there's the get us high part. Actually, that is again an oversimplification. I read an article sometimes since the beginning of the year that said that at this point we've like identified over 125 different active components in the cannabis plant. And so early on, we identified that THC is the one that gets us high, but there are a couple of others that also maybe even get it, people who get high higher than the THC component. But the point is, you can look at this. The point for our purposes is you look and see, here's the drug molecule key, here's the neurotransmitter molecule key, and the drug works. The drug does what it does because it fits into the lock that the neurotransmitter chemical key is built for. We'll be talking more about this in the next couple of presentations. So now I'm going to turn this off and I'm going to edit. I've got a lot of editing to do. Like I said, I hope it's not as choppy as the last one, but we've got time constraints and we'll see how it works.